Absolutely. There you go. So this is lesson 13. So for the first one, we'll take a look at the binding gels. What you do is you set up a line, one column basically for each of those DD-oxyribose. So the DD-GTP, DD-ATP, and so on and so forth. And then you assume that in each of those columns, while it's synthesizing, once it hits one of those DDG or DDA, it can no longer synthesize because nothing can be bound to that 3' end. And so then when you put them in the gel, the lighter molecules will go farther, and so they're farther down on the gel than the lighter ones. So when you go through the sequence of that sequence it gives you, you get a C at the bottom, then you get a T, then you get a G, then you get another C. Let's make sure I'm right here. Then you get an A, you get 2 T's in a row, then you get 2 G's in a row, then you get an A, a C, a T, and an A. And this is that picture there at the bottom. And you just figure that the heavier it is, the higher up at the top it goes, and that's a 5' to 3' from the bottom to the top. So then if we go to part B through D, so the first one, if you use the regular DNA polymerase, that would not work because the TORC polymerase, that is rated for a higher heating. And since we're about 95 degrees, DNA polymerase 1 would denature and would no longer be able to function. And so the whole process would just be shut down there. For C, helicase wouldn't be used at all because the denaturing at the beginning where we're at 95 degrees would separate the 2 strands, voiding any need for any kind of helicase, so it would still go forward as normal. And then for the last one, you suppose you added the DD-ATP but nothing else, you would get the DD-ATP would end, and that would be the only thing you'd ever see in the gel. It's just that brick. Everything else would be complete, and you wouldn't ever see it at all. So then for the second one, for the first question there, we have DNA synthesis. Step is done about 72 degrees, and annealing is done at 55 degrees. AMPR is a common selectable marker in the plasmid. Then you have the restriction enzyme, recognizes and cleaves the DNA molecules. DNA is positive. It will go towards the positive electrode because it is negative. DD-ATP lacks 3' hydroxide and the 2' hydroxide. The E there is transformation. This term used to describe the process where bacteria take up DNA molecules from the environment. And then blue-white screening, you have IPTG is introduced to express the lac operon, while XGAL is cleaved by the beta-galactoside to turn the E. coli cells blue. And there's the answers again on those.
All right, problem 3. So for part A, you're going to use the bottom part or the back part of your 3' to 5'. You use your TCG and so forth. And then your top one, you're going to use that first part, starting with GAGCC. And those are going to be your 2 strands. For part B, you need to count how many C's and G's there are. So in the first strand, 3 to 5, you're going to have 8/16, which is 50%. And you don't write that as 50%, you write it as just straight 50. And so you use the equation that it gives you for annealing temperature, and you get 62.3 degrees Celsius. For the second strand, you're going to get 9/16, which are going to be G or C, and use that equation again. And you're going to get 59.8 degrees Celsius for the annealing temperature on that one. For part C, you need to look at your, hold on, 3 to 5. I wrote these ones backwards. For your 5 to 3', you're going to have it's just going to stop at C because your first letter is G. And your 3 to 5, you're going to stop, or you're going to have a GTC, and it's going to stop there because you can't go any further with that C. It stops right there. And then part D, when we learned about DNA replication in E. coli, we learned about DNA AA and DAM. Why are these 2 proteins not included in the PCR experiment? The PCR is ultimately just trying to denature before replication, so you're not going to actually have DNA A and DAM in the PCR experiment. Problem 4, The first part is just asking for that little graph. And so you're going to have your reactions that are listed up here, and you're going to put those on top. These are going to be your base pairs, and you just put 50 where 50 is, and that's going to go for your BAM HI. And you're going to do that for each one of those. And then part B honestly takes the longest out of all of them because it's trial and error. But you're going to have your BAM HI, and left of that is going to be your 50 base pairs. Right of that's going to be your 150 base pairs. Eco RI is going to have 100 to the right of that. Your HIND 3 is going to have 100 to the right of that. And your BAM HI is going to have 500. And the way that works is HIND 3 is going to be 600, which it says up here, or 300, like it says at the bottom, right there. Part C, what are the predicted fragment lengths? You're going to have 50, 100, 150, and 500. 100 is going to be used twice, but I mean, you really don't need to write that. So there's your answers for that one. Question number 5 is asking for the steps that you use to generate cDNA from the Saccharomyces mature mRNA. And honestly, this one comes straight from the book. So I'll read it out to you real quick. To generate it, you need to purify RNA that is matured. Number 2, add a primer. Number 3, add reverse transcriptase and DNTPS. 4, add RNase H. 5, add DNA polymerase 1. Number 6, add ligase. To clone it, you're going to need your plasmid and chromosome DNA containing beta globulin, and they're going to be digested with the same restriction enzyme. Number 2, you're going to have your cleaved plasmid and your DNA mixed. And number 3, you need to add your DNA ligase. The blue or white screening, E. coli cells transformed with recombinant DNA. They're going to be transformed E. coli plated with ampicillin, IPTG, and XGAL, so you can see that zone of inhibition. 3, white equals it's going to contain your recombinant DNA, and blue is going to lack it. And then 4, you're going to use your restriction mapping to figure out what is able to grow and what isn't able to grow. And let me show those steps. All right, well, thanks for watching.